welcome to Nano. Today we're going to be discussing a great new paper released recently. Structure of a Janus Kinase cytokine receptor complex reveals the basis for dimer activation. This is done by a group out of Stanford, and the first author is Caleb Glassman. It's just a really, really important work as it's the first structure of the entire dimer, including both the pseudokinase and the kinase domains, and it's really, really great to be able to dive into this with my fellow nanomers, Carla and Daniel, application scientists, and Sheila here. And then our great recurring guest, Mike Bishop, who will be offering some insight into this as well. So I guess to start off, we'll just kind of do a little bit of background on JAKs, and just to kind of give an idea of what we're working with. And really, this is kind of showing where JAKs are and how they relate to cytokine receptors, of where we'll be seeing just a little bit of the box repeats that kind of bring these together here. But overall, it's just indicative of a larger receptor that will activate JAK kinases, and this will lead to the activation of the STAT pathway, which is implicated in a whole host of diseases, but focusing on cancer. And you can kind of see here how a lot of times there's sort of these heterodimers between two different JAK and including TYK2. And we can see these different cytokine receptors have different groupings of STATs that they'll activate. And that leads to different, phenotypic responses, whether it's, T cell proliferation, differentiation. We can also see that it's going to be involved in growth generally. And all these things, if they're misused or if things have been changed due to the cancer background, can overall lead to high levels of proliferation and high levels of resistance. And these are all things that being able to target JAKs capably will really allow us to go after these tumors differently. But any other thoughts or things you want to point out?
Yeah, so besides cancer, also there's implications for autoimmune diseases, I believe.
Yeah, arthritis.
Oh, absolutely.
I believe, right? Yeah.
But the really interesting thing here is we're actually looking at not a heterodimer, but a homodimer here of JAK1. And so what kind of occurs often with, tumor development is the formation of these aberrant homodimers that are able to be constitutively active, and there's a lot of mutations and things around these homodimers that we'll be able to explore today to kind of show this big concept of the paper, puts right in the title, the basis for this dimeric activation. And this is something that hasn't really been able to be explored without a great structure like this. And so we'll also be able to kind of think about other ways JAKs can be targeted now that we better understand another interface and see how druggable it is. Yeah. So Daniel, would you like to show us kind of that interface there and some of the interesting things we might explore?
Yeah, let's discuss a few structural features here. So the 7 domains of JAK1 form an extended structural unit whose dimerization is mediated by this pseudokinase domain, and it's enhanced by the oncogenic valine to phenylalanine mutation. That's exactly what we're seeing right here. And valine 657, which is this one here in front of me, it just enhances the shape complementarity of the pseudokinase dimerization, all this interface over here. And apparently it also facilitates the ligand-independent activation. So pretty interesting features here.
Should we look at the chemical interactions, do you think, in that pocket?
So yeah, here I'll just select each residue using our selector tool. Just being able to grab them quickly here. And then, we'll just kind of bring up over to the side from our stacks, the chemical interactions plugin. We'll run that, and it's pretty straightforward. So we have, we just need to indicate the protein structure we want to see the interaction with, and we'll just see the selected atoms and how they interact with one another here. So we'll just calculate that, and it should only take us, a little bit to be able to calculate these interactions. But
we can see down here, John, the different types of interactions we might see and how they're color-coded in Nanome.
Oh, yeah. We'll probably want to maybe look around and see if there's other interaction types other than our default 6 that we might explore. So, yeah, that's a great idea, Mike.
Yeah.
Let's take a
Some
look.
hydrophobic, perhaps.
Yeah, I'll add those once it finishes calculating to make sure we don't have any issues with that. And so what that can kind of show us is we can just be looking at distances, and this is another way of kind of understanding the differences here as we take a look. And so just kind of seeing the space here and seeing how these might be different. You said 657, right?
Yes,
Is
correct.
where the
You
mutation
want to
occurs?
maybe scale it up a little bit?
Yeah, let's make it a bit bigger.
Awesome.
So we can kind of
Awesome.
see the proximity of this residue to these other ones over here.
There.
Yeah. Kind of get an idea of some potential interactions and some of that space there while we can see it's about to update the workspace. So I'll just clear these measurements so we don't interfere with our perspective. Yeah, but a lot of good hydrophobic interactions. I'm sure if we brought up van der Waals, we'd see a similar set of interactions here. So there is maybe not a lot of strong interactions, but a lot of interactions nonetheless that filling that in there really, really helps with. Do we want to try to mutate the 657 phenylalanines to valine and kind of see what the difference is there?
Yeah.
Yeah, that's a great idea.
All right, I'll hide these. Duplicate them again. And we'll show these aligned to here, so we just have a nice friendly copy. So here we'll select 657. I'll make sure it's selected in the grouping we're interested in. And we'll just go into Edit and then into Mutate. Let me move this over to the side though, so we can kind of see it a bit better. And here we're able to see all the residues that we could choose from, and we have phenylalanine here. Within the nonpolar, we'll also see valine. And so I'll just select the valine, we'll switch it out there, and we'll just we'll hide the other phenylalanine for now. And
Maybe
we can
the
see
label
here,
too.
that's a good point.
Should we do the
Good
same
call,
thing with
Dana.
the other dimer? Yeah, the other
I think it's
phenylalanine
yeah.
over there?
I think it's a great idea. All right. So I'll just
All right, we're
once
almost there.
again- Yeah, almost. So we'll do valine. And from here, we can do a similar calculation for the chemical interactions again. And we can add those here. So this time I'll select this complex. And this time we'll just deselect these to begin with. And just look at the hydrophobic. And we'll calculate. And so from here, we can already tell once again, if we just bring up the measure tool, we can kind of look at these different distances that we're looking for. And if we remember correctly, to these closer residues, the closest atom was only about 4.8 angstroms away. And now, you can see that's been greatly increased. And interestingly enough, I just got a notification to say that there weren't interactions here between these. And if we reveal back our original structure, we can kind of take a look at why that is. So we can bring this in, and we can see just how much closer all of this is, and how really not missing those valines, having the valines turn to phenylalanines really bring about a much closer set of interactions, really fills the space that's needed for a lot of those positive hydrophobic interactions. And we can do the distance measurement to here to see that contrast again. Yeah, that's a lot closer.
Wow.
Right.
Yeah.
It's kind of
Makes
amazing
the whole
'cause you
difference
don't
with-
think.
Mm.
I was going to say you don't think of hydrophobic interactions as being very strong. We typically think of other interactions as being really powerful and strong, but so many of them and so well aligned is what's gluing these two dimer or monomers together.
Oh yeah.
Yeah, I guess the pi stacking ones are considerably stronger though than the average hydrophobic, which is a little less than a kcal.
Mm.
But it's interesting because these phenylalanines, at least for the measurements that you have, are slightly longer than what you would expect for pi stacking, which is usually around 3 angstroms, I think, 3 to 4.
I wonder if that's why there really I only saw one magenta line.
Yeah. I don't.
And then
Yeah.
You
there
could
were a
probably adjust
lot of
that,
just
correct?
pink
Or-
lines. Yeah, so maybe it went. That's too far to be considered enough to be a pi stack.
Pi
Yeah.
stacking, yeah.
I think so. Well, and
Good
the
point.
other thing to consider, while these are the key interactions that we spotted due to the mutation analysis, we can see pretty plainly, I'll just shrink this a little bit, that we have a lot of other interactions really proximal, that this is likely helping motivate and keep it together and oriented properly. But if we were to, select down this this beta sheet here, we've got 2 beta sheets that are more or less in parallel. And we can kind of see that while those residues are certainly important, we can see that there's actually, some hydrogen bonding that's going to be occurring from beta sheet to beta sheet across the dimer.
Yeah.
And so,
wow.
so it's
wow,
it's
that's
probably, the
beautiful. Look at that.
accumulation. Yeah, let's do a calculation of that. I'll just do the hydrogen bond so it's a
This
little
is
bit
like-
faster.
Yeah,
But yeah, you can
those
see
are
it
pretty
sort of
dreamy.
Yeah.
sewing together. Yeah.
Nice.
Oh yeah.
Very interesting. This. Wow.
Yeah, so this is interesting. So we've been looking at the dimer interface, and this is really important for mutant form of the enzyme, where it would come together here and stay in the activated form. But what people have been developing are inhibitors of the ATP binding site in the kinase domain. So I think, Jonathan, you do have a structure of one of those, just so we can see how far away that is. It's quite distal to this interaction we're looking at now.
So a useful figure from the paper that I really, really like to kind of help emphasize this, it shows the cytokine receptor here, transmembrane. Into the box repeats, we see they're rich with prolines to prevent alpha helix formation. But then we see down here the pseudokinase, which we're seeing from their research, really proving that this is sewing this together. And then, like Mike mentioned, we now have the kinase domain a bit lower, which we'll see right here. And the really interesting thing that we kind of have to show you is I'll hide these structures here for now, but we have an aligned structure.
Oh, yeah.
Oh, do we? Doesn't look aligned. Is this the right one, Carla?
The 6AAH should be correct, but we did duplicate the original, remember?
This is the... Oh, wait, this isn't the one that was aligned. This is the one that is aligned, I think.
Yep.
Let me show the ribbons. Does it not have ribbons? Oh, okay, there we go. Okay, there we go. Sorry about that. And then we'll hide the ligand. We'll just cut all this out. So here we can kind of see the original structure that was shown. I'll hide this briefly. Had an ADP, molecule right inside here, and this is what would fit into, the kinase domain to be able to undergo phosphorylation on its targets, the stats. But if we hide this ligand and instead we kind of show our alignment here, you can see a specific inhibitor of JAK1 and how it fits into here. But yeah, anybody, we can make it a bit bigger and discuss how this is fitting in and how maybe we get new insights from the total complex.
So can you point out the DFG loop? I'm just curious about the orientation of the activation loop to see what conformation of the kinase it is.
So
maybe they mentioned that in the paper.
Well, let's I'm seeing that's a DG. Let's see if we can find the DFG loop. I mean, my guess is I'm just kind of going here. We'll just check the sequence and see if we're...
This almost looks inactive to me, but I don't know.
No.
It could be wrong.
Let's see here. We can look through here and we should be able to see...
There it is. Yeah,
Okay,
Meg found
that
it.
seems likely. Let's just bring up that as sticks and... Okay.
Yeah, you were right.
Oh, I was right. Yeah, I second-guessed myself because I saw the glycine over here and was
Yeah.
like, oh no. Okay, yeah, it's just a little bit further up. But yeah, we can see the DFG group here. And we can see the C helix off to the side. And as Carla was mentioning, if you're not familiar with kinases, but essentially in order for a kinase to be active, it needs a DFG in C helix in, kind of brought into here. And we can look at the along the hinge, we'll be able to get to a gatekeeper residue.
This is exciting to see, and there's several JAK inhibitors that are on the market and others that are in the clinic now to treat a number of diseases like we talked about earlier. But when you think about it, Janus kinases do a lot of things in the body in disease states, but also in healthy tissues. And this enzyme here is going to inhibit JAK in any tissue where it should be, transferring a phosphate. So imagine with this dimer structure now, which is a type of mutant that's activated all the time when it's dimerized. Could we use this to try to design a molecule that would be targeted towards that mutant state, as opposed to something that would bind here and block all JAK kinase activity? It's an interesting thing that this dimer structure brings to us, is possibly a new way to treat diseases driven by these mutants.
Absolutely.
Right. And in terms of selectivity, there are over 400 kinases that all bind ATP. So that ATP site is similar for everything. But that dimer interface is likely to be fairly unique. Or that would be a hypothesis to go after.
Well, and even just kind of getting even a little bit crazier, perhaps, we obviously, looking at a small molecule here, we see how it can get in and really deal with the active site directly. But even just this entire dimerization, structure, this dimer here of a homodimer that isn't really, natural. This JAK1, it's not one that's going to be typical. Finding ways of maybe making a PROTAC that's able to recognize the interface that is specific to the disease state. Small molecules, that break this up might be able to prevent it in one way. Degradation might be able to go in a different way. And honestly, I don't see a reason to not go for every option and explore each possibility until we find the best one. But it is just different, having this space really, and being able to visualize it capably and see those differences and quickly evaluate key spots like the beta sheet to beta sheet are all things that visualization can really amplify.
Yeah, disrupting the interface would be definitely a good strategy.
Well,
but yeah, of course, degrading it is just another alternative. Yeah, it's a very hot topic, right? Degrading the proteins right now.
Do we understand why the formation of this dimer leads to an active state? Or do we just know that it does?
We do know a bit about it. So something to kind of know about this overall. We have the firm SH2 domain. And with a lot of SH2 domains with kinases, they are autoinhibitory. And their job is to more or less hold part of the kinase domain, and kind of prevent it from being able to be accessed and activated. And oftentimes the SH2 domain will get phosphorylated, and that will allow for conformational changes to sort of activate or release the kinase domain into an active state. And so one way that people have kind of thought about this is that part of that kind of association can be somewhat disruptive. And we can kind of see here that the pseudokinase domain being able to come together in this way allows for other conformational changes to occur to release the kinase domains and to kind of reduce the ability for the SH2 domain to be able to capably control that anymore. And so you can see that right here of where in 4-OLI, if we wanted to open up that structure, we could see this sort of folded in state. And what happens instead is the kind of contact points here that really hold it together, they get mutated, making this somewhat floppy, and then it gets fully activated by this dimerization that maintains this conformation. And so, yeah.
That makes sense. So it's blocking it curling up in on itself like that.
Yeah.
How it is in this structure.
Exactly. And you can kind of see down here some of these same parts of it, and seeing how mutations in this interface, it's really, really key here. And, how this kind of all comes together through associating both of the cytokines, which can also dimerize themselves, which keeps them in close proximity. But like Daniel already mentioned, if we can allow for dimerization without needing the cytokine receptor homodimer to drive it, you can really exactly get that ligand-independent dimerization that can, like Mike was pointing out, it just becomes active when it shouldn't be active, and that's the real problem. And making sure that you don't target, sites that can be produce poor off targets, like Sheila was mentioning, but there's a lot of kinases. It's all things to consider the structure like this. Great question, Sheila. I'm glad you asked it, because that's a really important point to the whole paper.
Right. Absolutely.
I was just wondering if we actually had a structure in the space of the inactive conformation.
Oh, yeah, we can load that in real quickly. It gave us the PDB ID, so we can do 4-OLI. And we can kind of look at how this looks different. And so here we can see, I'll hide the everything but the ligands, so we can kind of track where the where all this is. But we can kind of see here, this is kind of our kinase domain, this is our pseudokinase domain, and this is the SH2. And if we just kind of place it over top of this here, we can kind of see how, these different areas, more or less, like if we aligned it, it might be easiest to align it, visually right here, where the kinase domain is. You can see how those ribbons are all overlapping. And you see there's a huge conformational difference everywhere else.
Yeah, it's way shifted. Oh.
Yeah. It's missing the... Yeah. And you can kind of see, once again, in this figure, we don't have the SH2 domain. This is just the pseudokinase and kinase. And, yeah.
So this is the pseudokinase, and the kinase is here, right?
Yeah.
And then our dimer, the pseudokinase, is way over here, right?
Yeah, exactly. And we can rearrange it so that we...
That's amazing.
We try to rearrange it by this one. But that's a little bit harder, considering how different the conformation is. But yeah, you can see, it's a big difference. But yeah. All right.
Really
well,
cool
I feel.
Thank you.
Thank you all for joining us. Really, really great topic. Really gave us a lot of interesting ideas about drug discovery and targeting difficult, structures like this one, but how many new opportunities as we understand the structure differently.